Let's take a look at transcription and translation and also replication. Let's first consider what these processes are for. So transcription and translation, that's the process that you need to follow in order to make proteins. So it's protein synthesis. Transcription is the process by which we take DNA and transcribe it into messenger RNA, and translation is the process by which we go from messenger RNA then to protein. Replication is the process of making a copy of the entire genome of an organism in preparation for cell division. So you need to copy everything start to finish. In transcription, you only copy what you need for one protein product. So let's start out here with transcription and translation. There's something called the central dogma of molecular biology, and I would like you to know this. So this is really the big picture here for transcription and translation. And you go from DNA to messenger RNA. So I should say messenger RNA. Put that little M there, messenger RNA. From DNA to messenger RNA, we're going via transcription. So we will copy one recipe, if you will, one protein, what you need, the information you need to make one protein product. We're going to copy that from the DNA. There's a lot involved in this, and to get a finished mRNA product is actually pretty elaborate. It's a pretty long process. But then we send the messenger RNA. Once it's done, we're sending it out from the nucleus to the cytoplasm, and then the ribosome will attach to the messenger RNA and will do the job of translation. So that's the second part. This is the process by which we go from messenger RNA to a finished protein product. The whole goal is to make proteins. And of course, proteins do everything in cells. So if you want to do any kind of metabolism, you got to have proteins. Okay, so starting out with transcription. The goal of transcription is to make messenger RNA. So we need to take the original DNA that's in the nucleus and change it to a more portable molecule, which is the messenger RNA that then leaves the nucleus, goes to the cytoplasm for translation. There is this relationship of one gene to one polypeptide. So one gene codes or contains the message for one polypeptide chain. So this here is your protein, and the gene is the instruction for how to make this protein on the DNA. We have this mediator molecule that we need, and that is the messenger RNA. So we go from the DNA to translate the information that's on the DNA into a protein. We need to have a mediator molecule that's transportable, because the DNA cannot leave the nucleus, and protein synthesis takes place in the cytoplasm. So we need to have something that carries the information from the nucleus to the cytoplasm, and that would be the messenger RNA molecule. So let's take a look at this here. This is a pretty nice overview picture that shows you what we need to do. So here in the nucleus, we have the DNA, a double strand in red right here. Via transcription, we will make messenger RNA. So we'll make I refer to it as a cheap copy. It's a quick copy. It doesn't last. It's not built to last. It's not meant to stay for a long time. It just carries the information from the DNA to the cytoplasm, and we're using this information molecule. So it's basically, think of your DNA as a cookbook that contains all the recipes that you know how to do. If you just want to make one dish, you wouldn't be and you want to give somebody the recipe for one dish, you wouldn't be copying the entire book. You just copy that one page that has the recipe and give it to your friend or whoever wanted it. So in a similar sense, the DNA is the permanent molecule that stays in the nucleus, and the messenger RNA just carries the information for one protein product out of the nucleus to these nuclear pores into the cytoplasm, where then the ribosome will read the information that's on the messenger RNA, the ribosome in red, and then translates it into a protein product. So they're in green. So that's the translation process. The ribosome literally is a translator from messenger RNA to protein. Okay, so here a little bit more information on transcription. The DNA, as you know, is a double strand. And RNA is a single-stranded molecule, but still the base pairing rules apply. Now in DNA, the base pairing rules are A with T and G with C. In RNA, we don't have any T, so we're going to replace any time where we needed a T, we're going to replace that with a U, with a uracil. Okay, but other than that, as long as you know the coding strand, since we have 2 DNA strands, it's kind of tricky to know, but we don't get into details like this. So well, the coding strand will be copied using the base pairing rules into an RNA molecule. And the initial RNA molecule really needs to be further processed to be of any use. So let's take a look at all the different types of RNA that you should know about. So of course we talked about the messenger RNA, and there's a precursor to that, so that's called pre-mRNA, but then it gets processed to make the mature or the ready to use messenger RNA. For the translation process, we also need ribosomal RNA. That is the ribosome itself. So the ribosome is composed of ribosomal RNA, that's abbreviated rRNA. And then also necessary for the translation process is a small molecule. It's called transfer RNA or tRNA. So those are the RNAs that you need to know about. The messenger RNA was its precursor, pre-mRNA, then the ribosome itself, the ribosomal RNA, and then we need a mediator molecule that sort of does the translation job in conjunction with the ribosome, that's transfer RNA or tRNA. And here a little bit about pre-mRNA. The thing is that the DNA of eukaryotes has a lot of non-coding sequences, and they are part of the DNA molecule. Think of it as an encrypted message. It has part real message, and then there is some mumble jumble that's embedded into it that sort of is a distractor, and it doesn't code for anything, but it makes it hard to read the information. So it's sort of an encrypted message that is in the DNA. And the parts that don't code for a protein product are called introns. And the initial transcript, so when you're copying the DNA into RNA, these introns, they will be copied along with it, but they need to be cut out. And so here you see the process of intron splicing, we call that. So these introns, they are this mumble jumble message that we don't need. We need to cut it out. So we need to get rid of these pieces. Here, these dark blue pieces. They were part of the initial transcript because we have to copy everything that's part of a gene at first, but then we need to cut out the part of the information that we don't need. Those are these introns, and that's called splicing. So introns are regions of non-coding DNA within a gene that need to be cut out, and it's a pretty elaborate process, but we'll just keep it short here because that's not really the point of this lecture. So the exons, that's the message that will be expressed as a protein in the end. So that we need to keep. And so we just need to cut out the introns. And then we have our mature messenger RNA that we can then send out to the cytoplasm for translation. So translation is the actual process of making the proteins. So we need the messenger RNA. It needs to get out of the nucleus to these nuclear pores, and then it needs to attach to a ribosome, or multiple ribosomes. Here we're talking about polyribosomes. So multiple ribosomes will attach to that messenger RNA and start reading the information and translating it into protein. So the actual translation process is the process of the ribosome reading a triplet code on the messenger RNA and then translating it into amino acids that make the protein. So here is protein synthesis, sort of in a nutshell. We're starting out here with our double-stranded DNA molecule. The DNA coding strand is the one that will be then transcribed into messenger RNA. And so you can see here the top part in purple is DNA, the coding strand, and then the messenger RNA portion will be with the base pairing rules. That is what you then use to make the messenger RNA from the DNA coding strand. So let's take a look at this. So a T on the DNA would be transcribed into an A in the messenger RNA. And then here base pairing rules, an A on the DNA. Normally, if we were to just replicate and make more DNA, then this would be a T. But since we're making messenger RNA, this is a U. So make sure anytime you see a U, you know it's RNA. And then C with G, these base pairing rules, they stay the same, but here again, A with U. Now, T on the DNA will be base pairing with A. So I do expect you to know how to go from DNA to messenger RNA. That's probably going to be something along those lines on the first exam, so kind of be ready for that. It's not going to be difficult, but you do need to know what the base pairing rules are and how you go from DNA to messenger RNA. And we're not going to deal with any splicing or introns and something like that. Nothing complicated. We'll go directly from DNA to messenger RNA. And then we're going to go the actual translation process. We'll now read the triplet code, and that is known as the genetic code. It is universal across all forms of life on planet Earth anyway, with very, very, very few exceptions. There are some, but none of our concern right now. So the universal genetic code will be used to translate the triplet code on the messenger RNA into the amino acid language of proteins. So we're going to take the first triplet. A triplet is 3 nucleotides on the messenger RNA. We're going to start out with AUG. AUG is called codon number 1. That's the start codon, and it translates into the amino acid methionine. And then here we have GGC. That's codon number 2. So these 3, this triplet right here is codon number 2, and it translates into a glycine. How do we know? Well, there is sort of the dictionary of the ribosome. That's the universal genetic code, and I'll show you that in a sec. But you do need to know how to use the genetic code to do the job of a ribosome. And you will always be given the genetic code, of course, if you are expected to translate something. But you do need to know that you need to take a triplet on the messenger RNA, triplet by triplet. So the next codon would be codon number 3 or triplet number 3, and that UCC translates into a serine. Then here is AUC, that's the next triplet. Codon number 4, that's an isoleucine. Then we have GGC, that's codon number 5, translates to a glycine, and so forth. So we have this one has a blank in it, so obviously if this is G, then this should be C, so we can put that in here. And I might give you a DNA coding strand, expect you to do the base pairing rules and transcribe it into messenger RNA, and then give you the genetic code and have you translate a few triplets into the amino acid sequence. So here we have GCA, and that's codon number 6, this triplet, and translates into alanine. And then here we have another GCA, and that will be another alanine. So a little practice and you'll get the hang of it. It's not that difficult. So here, one more time to make sure that we're good on this. We have here the DNA strand. So we should just for fun transcribe that into mRNA, since it's not shown here. So you can practice with that. But TCA would be transcribed into AUG. Then this here AAA is going to be U. Remember, no T's in mRNA. And then we have CCA, that will be GGU. And then here we have UCC. This will be GGU. And then we have UGG. And here we have CAU. And then UC. We're done. that's messenger RNA. So you should practice a little bit with that. Maybe you can erase that and kind of work with that and practice that. And so then we go to the genetic code and we find that AUG translates to methionine, UUU translates to phenylalanine, GTU to glycine, and so forth. So let me show you how we're using the genetic code. I have the genetic code a few slides down. There it is. So here, I told you earlier, the AUG is the start codon, and it happens to be this one right down here. So AUG is our start codon. There we go. And let me just go back. I'll show you a little bit more about the genetic code, but let's just do these couple slides here. So for protein synthesis, each mRNA contains hundreds of nucleotides. Depends. On average, I would say that's true. There are some messenger RNAs that are really, really short, only translating to a few, a short peptide, let's say 9, 10 amino acids. And then some are huge. Some are really, really large and contain even thousands of nucleotides. So it depends, but on average, so a few hundred nucleotides, and the base pairing rules apply, and then each on the messenger RNA, a codon is defined as a triplet of nucleotides. So 3 bases or 3 nucleotides form a triplet, and that's sort of a code word for a specific amino acid. So it basically works like translation, like you would using for a language. Okay, the genetic code is how DNA codes for an amino acid sequence. There are 4 bases in DNA and 4 bases in RNA as well, so AGCU in RNA, and they make triplet words. So if you have a 4-letter alphabet and you're making triplet words, and you have 4 times 4 times 4, so 64 possibilities of making triplet words. We only have 20 amino acids, so there's not a one-to-one match. In other words, we have more possible triplet words, code words, than we have amino acids. So there's some repetition, or we call that redundancy, in the genetic code. So redundancy of the genetic code means that there's sometimes, or actually a lot of times, there's more than one code word for the same amino acid, and you can see that in the genetic code. So here again, here's the genetic code, and you can see exactly that. So here we have all 64 possible triplet words. They're spelled out here. The first letter of a triplet word is in red, the second letter is in blue, and the third letter is in green. So I've kind of color coded here for you. So for the first, second, and third base or nucleotide. And you can see that phenylalanine has 2 code words, UUU and UUC. Leucine has 6 code words, UUA, UUG, and all 4 of these. Methionine has only 1. And you can just muse over this. You don't have to memorize this. This will always be given to you. But this is the universal genetic code. And remember that we're using messenger RNA triplets. Okay, that's important. mRNA triplets. And there are 64 of them. Oops, 64. There. So now these 64 mRNA triplets, this is the genetic code, shows you how they get translated into amino acids for protein manufacturing. One more thing here, there are 3 code words that don't code for any amino acid. They're known as stop codons, because once we hit one of these stop codons, then translation will stop. So here are these 2 are the stop codons, and then here's another stop codon. So 64 minus 3 is 61. So we have 61 code words that code for 20 amino acids. So you can see this is still not a one-to-one match. So we have roughly, on average, 3 code words that code for the same amino acid. Okay, now here is another way. This is what you find in your book. I'm not a big fan of this kind of chart here. I like the full genetic code. So this one here, this is my favorite. And if I put it on a test, you will get exactly this chart to look at for translation purposes. But here is another way of spelling out. This is the DNA triplet, so a triplet of nucleotides in the DNA. This would be the RNA triplet or RNA codon, and this is the amino acid that it translates into. And you can see the AUG here is still the start methionine, the UAC is a stop codon and whatnot. So I just like the other chart a little bit better, but the information here is the same. Okay, now let's talk a little bit about transfer RNA. So transfer RNA, that's the mediator molecule, looks like a little clover leaf. It's made out of RNA. That's why it's called tRNA. And the T here, the transfer RNA, comes about because this is your mediator molecule. It carries on one end of the molecule an anticodon that matches with the code word on the messenger RNA. So here, down here is our messenger RNA, and this here is our code word that we're currently reading on the ribosome, and it's UGG. Now we need the UGG. If you take a look up here, so where do we have UGG? So on the genetic code, you take a look, and UGG is right here. So that's going to be a tryptophan. So we confirm that. And so here comes the transfer RNA. It carries a tryptophan on the other end, on the opposing end. So the anticodon is here that matches with the UGG. On the mRNA, we know that's going to be a tryptophan, so we need a cloverleaf shaped tRNA molecule that carries the anticodon that matches this codon here, the code word, and it carries a tryptophan. And so the job of the transfer RNA is to establish this code word base pairing with the anticodon to the codon. So this is according to base pairing rules, and then bring along the correct amino acid. So that's the job. And then we're going to do this multiple times, and that's how we're going to form polypeptides.